Hello and welcome to this webinar titled Advanced Process Development Using Automated Microbioreactors Shortens Timelines and Provides Process Solutions for a Rapid Scale-Up, hosted by Biopharma Asia Magazine and presented by Dr. Jincai Li, Vice President of WuXi Biologics, and Dr. Liang Tang, Vice President of WuXi Biologics, and Sunil Chhatre, Sartorius Stedim Biotech. My name is Stephen Edwards and I will be your moderator. Now, please allow me to introduce our first presenters, Dr. Jincai Li, Vice President of WuXi Biologics, and Dr. Liang Tang, Vice President of WuXi Biologics. Dr. Jincai Jimmy Li is currently Vice President of WuXi Biologics. Dr. Li has over 17 years of experience in biologics process development, scale-up, and cGMP manufacturing. His group contributed to over 40 IND submissions, both outside of China, as well as BLA submission works. Dr. Li joined WuXi Biologics in 2011, helped build and led the cell culture process development and pilot plant production group, and then managed the cGMP drug substance manufacturing facility. Before joining WuXi Biologics, Dr. Li was a cell culture process development group leader at Biotech Inc. Before that, Dr. Li also worked at Tang Inc., where he led the manufacturing sciences group supporting pH3 production of an MAB product, and at Diversa Corporation, now BASF, in charge of process development for multiple recombinant enzyme projects. Dr. Li holds BS degree from Tsinghua University and PhD degree from University of Maryland, Baltimore County, majoring in chemical and biochemical engineering. And Dr. Tang is currently working at WuXi Biologics as a Vice President of Biologics Development and Manufacturing. Apart from leading cell culture process development function in product delivery and new technology development, Dr. Tang also serves as a CMC leader and provides leadership in development and manufacturing alliance management, product development and manufacturing strategy for selected projects. Dr. Tang was trained in protein biochemistry and molecular biology from McGill University, Canada, and had a postdoc training at the Biochemistry Department, Imperial College of Science, Technology and Medicine in London, UK. Before joining WuXi Biologics, Dr. Tang works as a function head of a molecular biology, cell culture and protein expression for Bio-US at San Francisco, USA. He worked over 14 plus years at Biopharmaceutical, participating in many protein therapeutic projects in CMC, supporting bioprotein therapeutic products in the market, such as Gamunex TM, Cogonut, FS, and Betason, and also actively involved in the research of novel protein therapeutics in hematology and ophthalmology, supporting the biobiologic therapeutic pipeline. I will now hand over to our first presenters. Welcome, Dr. Li and Dr. Tang.
Okay, good afternoon.
Hi, thank you, Stephen, and thank you everyone for joining the webinar. This is Jincai Li. I will start with the first part of the presentation, and then Dr. Tang will follow up on the second part of it. So before I start, I would also like to acknowledge some of our colleagues who put in a lot of effort, both in terms of the microbioreactor work as well as preparing for the presentation. So the names are listed here: Xiaoyuan Xie, Weiqiang Song, and then Huilin Zhu. So I'd like to start with the outline of this presentation. We will go over the microbioreactor features and their advantages, and also share our thoughts on the needs and the bottlenecks in process development. And then we will spend a little bit more time to share the application of microbioreactors for upstream process development. We'll start with an overview and then share with some case studies. And that will be ended by the conclusion as well as some perspectives. So we can see that current workflow for bioprocess development has a few of the bottlenecks. The benchtop bioreactors has limited throughput, and also because of the intense demand for labor setup, sample handling, and clean down, they are also prone to human error as well as inconsistency. So this call for a need, for more good scale-up, scale-down models that can provide representative data for the eventual larger-scale manufacturing. So in overall, we see the increasing need, for faster, higher throughput, and cost-effective approaches to process development. When we survey the landscape of microbioreactors, we can see that they can fall into two main formats. One is micro-titer format, and the other is a small stir tank format. For the micro-titer format, the throughput can go up to very large numbers. Typically, the volumes are milliliters or even smaller. The good products mostly have online monitoring of dissolved oxygen, pH, and temperature. Because they are micro-titer based, they agitate and mix by the orbital shaking. So this is not very representative of a large-scale bioreactor, and thus can pose some challenge for scale up. The second common format is the stir tank based. It offers decent throughput from 12 to 48 bioreactor formats. The volumes typically are a little bit larger, go up to 250 milliliter. Virtually all of these products have online monitoring of DO, pH, and temperature. And an important distinction from the micro-titer is that those stir tank-based formats mostly have air sparger and impeller for agitation and mixing. And because they are stir tank-based, they tend to offer a more similar geometrical design when compared to large-scale bioreactors. Here we show a quick overview of what's available on the market. It's not meant to be an exclusive list, but we feel that it offers a pretty good summary of what's available. So on the left side are the micro-titer based formats, and then on the right side are the stir tank based formats. And you can see that the volume ranges as well as the throughput. Another important feature is the degree of automation. So we gave a two stars or 3-star mark based on the degree of automation. So a few of those offers a pretty good degree of automation, including the Ambr as well as the SimCell. But SimCell has since been discontinued from the market. So virtually all of the microbioreactor products have a few features. All of them are miniaturized by definition. As mentioned, some of them, especially the stir tank based, have a similar geometrical design than larger scale bioreactors. Automation has been a good feature and important advantage. Some of the more advanced products offer automated sampling, feeding, as well as monitoring and control. And we also see the trend to have even more integration with, for example, offline analytical assays, as well as potentially linking to downstream development, for example, high throughput, small volume purification, as well as analytical assays. So a quick summary of the advantage of microbioreactors. Offer semi-high to high throughput bioprocess development. The integration with automation can help to reduce human errors. And because of that, as well as the mass production of the especially the disposable bioreactors, we actually see remarkable consistency and reproducibility from a bioreactor to bioreactor, as well as from batch to batch. Also, with the bioreactor features, they provide very representative data for scale up. This is an important advantage because it helps to reduce risk, especially during the early stage process development. Oh, okay. And then as an important result of this is the time saving because of the throughput and because of the automation. Cost-wise, we also see that it is very cost-effective. So I'd like to next share with you in terms of the applications of microbioreactors at WuXi Biologics. So first, we start with a typical overview of a process development scheme. With typical benchtop bioreactors and without the use of microbioreactors, the screen basically shows how it's typically done. So we will start with clone screening, start with about, let's say about 40 clones. Those are typically done in shake flask or spin tubes. And then we pick the top 3 to 5 clones and then move to bioreactor for evaluation. That's followed by process optimization, which typically will narrow down to 1 at the most 2 clones, and we'll do at least about 2 rounds. Once the process is deemed optimized, we will then move to confirmation and scale up to 15-liter bioreactors. So a typical process development without any major challenge will take about 24 weeks, 2 to 3 FTEs for each molecule. So we see a tremendous opportunity for microbioreactors to help reduce the timeline as well as save the labor cost. And next, we will share some of the implementations at WuXi. So at each of the stages during the process development, we have actually done a lot of projects with microbioreactor. For example, at clone screening, we have had more than 30 projects where the microbioreactors can help facilitate clone screening by providing a predictive bioreactor model, especially with pH and DO control. Further moving down, in process optimization, we have over 20 projects where the microbioreactors can basically replace benchtop bioreactors for process optimization. We also have multiple projects where the microbioreactors can actually serve as a good scale-down model to help scale up to the larger scale and GMP scale manufacturing. In addition, we have started to using microbioreactors as a scale-down model for some of the late-stage optimization as well as process characterization studies. So here, next, I would like to share a few different scenarios of how it's being integrated into the overall workflow. This is the general workflow with traditional benchtop bioreactors. So if we, for scenario 1, if we use microbioreactor to replace the shake flask and the spin tubes for clone screening, then what we can achieve is essentially combining the first 2 stages into one. Because by using microbioreactors, you are already screening your clones in the bioreactor environment. So that can save about 4 weeks of time. For scenario number 2, we can further implement microbioreactor to process optimization. So you would start with using microbioreactor for clone screening, as well as using microbioreactor for process optimization. And that can save potentially up to 8 weeks in overall process optimization work. Because of the high degree of automation, it also helped to reduce the workload quite significantly. So with that, I will hand over to Dr. Tang to share a few case examples.
Yeah, good afternoon. So I'm going to introduce some case studies from WuXi Biologics. The case study 1 is exactly totally mentioned earlier. It's combined the first clone screening and also process bioreactor evaluation steps into one stage and doing in parallel different microbioreactors in the process. And then we cut the time shorter to enhance to improve the process in the process development. And then from the slide here, you can see using in parallel two microbioreactor, we compare the results of coming up from these two microbioreactor A and B. Their results are very similar. If you look at the dash box there in terms of VCC and also in terms of expression titer, they are very similar. And then if you look at the viability and expression titer again in the graph format, they are compatible. So using different microbioreactor, we can get a similar results in terms of process development. So as I mentioned this microbioreactor provide a good method, a good tool for us to use in the cell culture process development. And the case study 2 is using a microbioreactor in a scale-up for a pilot production or production at the pilot scale. And here it shows we use microbioreactor in the process optimization and then getting into sub 200 liter pellet plant expression. And then you can see we used microbioreactor or 3 liter in satellite runs just to use as a backup for the 200 liter scale expression. And then for the 15 liter expression here in a traditional bioreactor was mainly due to more material were needed for process development, such as for the formulation development or the downstream process development. So at the current stage, we're still working to find how can we bridge microbioreactor to provide a good enough material for the whole process development steps for formulation, for assay, and also for purification. That is the result showing the scale-up comparison. And then we have here microbioreactor, a B type of expression titer, SEC result, and the purity compared to 3 liter or 15 liter benchtop bioreactor, and also compared to 200 liter sub single-use bioreactor. They all come out as a very similar result. Doesn't matter in the titer or in the SEC analytical data or purity. So the very good scale-up prediction when we use the microbioreactor in our process development. And then the case study 3 shows our experience in work on the biosimilar biologics, which tend to have a target the QPA for a target for the molecule, and in this case is a GOF, and then for the reference material was 69. And when we use the traditional glass bioreactor, 7 liters glass bioreactor in the system, and then we found later during the scale up at the 200 liter sub expression, we noticed this GOF percentage had a big difference between a glass bioreactor and a 200 liter sub bioreactor. And even when we try it in the microbioreactor, we noticed the microbioreactor percentage of GOF actually is very similar to 200 liter sub bioreactor, because we think that's because they are both of them are single-use system, and then maybe indicating microbioreactor in this case a better scaled down model for 200 liter sub. With that information, we did some additional work and using microbioreactor to do a fine-tune of culture condition and including the feeding adaptive different amount of the feeding condition, and then we actually be able to identify the condition which will provide us the molecule which meet the target in terms of QPA. After fine-tune, and then you can see from these results here, if we compare the optimized process MBRA, the number 68.7 actually is very close to the reference GOF percentage, and which is different from the old process. And then that condition, we will be able to scale up to 200 liter sub and get it very close GOF percentage at the 71% plus minus 2 in that range. So that is one of the important application for microbioreactor. In this case, which has a better scaled down model for the 200 liter, and then at the same time, microbioreactor with the semi-throughput ability allow us to screen for different conditions and then to identify the best condition for the optimize the process in order to scale up successfully scale up to 200 liter sub expression. So in summary, using microbioreactor, we can speed up the process in the cell culture process, starting from clone screening, process optimization, and then even process log, scaling up or scaling down evaluation, and then also some other applications using a microbioreactor in order to streamline the process, our cell culture process from DNA to IND submission. So this is a faster, more high throughput and cost-effective process development, and allow us to do a good work, provide a high quality of product, and then robust process for biologic development. With that, and then we'd like to thank teams, WuXi Biologics CCPD team, and especially thanks for the microbioreactor staff working on that project, and then also thanks CLD and other department teams, and thanks WuXi Biologics project team and the client collaborators and the vendors. And also like to thank Biopharma to organize the opportunity to to present the data. Thank you.
Thank you, Dr. Liang and Dr. Tang. Once again, I'd like to remind our viewers that there'll be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation via the questions tab located underneath the webinar screen. And we will go through these at the end of the presentation. Now, please allow me to introduce our second speaker, Sunil Chhatre, Sartorius Stedim Biotech. Sunil Chhatre has been a bioprocess product specialist at Sartorius for over 6 years, providing technical application support across the full range of cell culture and microbial fermentation products within the Ambr 15 and 250 portfolio. In this role, he has conducted many pre-sale trials and post-sales training at a wide range of commercial organizations and academic institutions, as well as providing on-site and remote support to a number of other specialists within Sartorius. His primary focus these days lies on the market in Asia, the Far East and Australasia, but additionally, he has worked extensively in Europe as well. Before joining Sartorius, Sunil studied the biochemical engineering at University College London in the UK, where his research focused upon the development of small-scale models for a range of biopharmaceutical operations with a particular emphasis on chromatography. I will now hand over to our second presenter. Welcome, Sunil.
Thank you very much. Good day to you all. So, following on from the last presentation, this one is going to focus in particular on one specific type of microbioreactor system, namely the Ambr platform, which, as Stephen mentioned, is a set of systems that we've developed here at Sartorius for small-scale operations, primarily focused on screening in the very early stages and then later on developing additional products which can assist with development, characterization and optimization, looking both at cell culture as well as microbial fermentation. And in this particular presentation, I'm going to start by just trying to put the Ambr platform that we manufacture into some sort of focus and look at it in terms of the other Sartorius upstream products. Then I'll move on to talk about the Ambr 15 cell culture system. This will form the majority of my presentation, and I'll hopefully go through the basic theory that describes how this platform works, as well as showing you a few pieces of data from some of our customers. I'll also spend a little time talking about one of our other products, which was developed a little bit more recently, namely the Ambr 250 high throughput, and I'll also be mentioning in particular one of the most recent developments for this one, namely the addition of a perfusion option for the Ambr 250 high throughput. Okay, but as I said, I just wanted to start by setting the scene, so to speak. So the Ambr platforms are shown at the top row of this slide, the first three products. They sit very nicely within the full range of the Sartorius upstream portfolio. So in the top left-hand corner, you can see the Ambr 15 cell culture system. We have units which offer either 24 or 48 vessel capacity. Further on through development and later on, we have other products such as the Ambr 250 modular and the Ambr 250 high throughput, which are more intended for later stage activities, perhaps later stage screening, as well as process development, characterization, and optimization. And those three feed in quite nicely into the rest of the Sartorius portfolio, offering reactors at bench scales of up to about 10 liters, and then going all the way fully up to manufacturing at 2,000 liter scale. But as I said, I wanted to focus in particular on the Ambr 15. You can see on this slide we actually have two different versions of the 15, one for cell culture and one for microbial fermentation. They look very similar and operate in many ways in similar ways. They both have very small working volumes, in the case of the cell culture system between 10 to 15 milliliters, whereas for the microbial system we operate slightly smaller, between 8 to 12 milliliters with 2 to 3 mils worth of gas holdup. In the case of the cell culture system, we have two versions, as I mentioned, one for a 24 vessel capacity unit and one for 48 vessels. The Ambr 15 microbial version is only available for a 24 vessel capacity. But both units are used to carry out the same basic types of activities, i.e. very early stage upstream work. cell line screening, media screening, maybe a little bit of early stage process development. For the microbial fermentation, similar strain screening, media screening, and again, looking at perhaps trying to do initial amounts of work for, say, the right type of pH, the right type of DO, maybe the right type of temperature shift, and so on and so forth. So these are our early stage product offerings. Later on, when, as I said, we move further into the development, we have two different types of product, which we call the Ambr 250. One is the Ambr 250 high throughput, which you can see on the left. This is used for both cell culture or microbial fermentation. Capacity is slightly bigger, so the bioreactor in this case runs between 100 to 250 milliliters, and the units are available as either 12 vessel or 24 vessel capacity hardware. As I mentioned, this is predominantly intended for later stage development, optimization or characterization activities. We also have a slightly smaller unit called the Ambr 250 modular, again used for both cell culture and microbial fermentation. The same basic type of reactor running between 100 to 250 milliliter working volume, but in this case the throughput is deliberately made slightly smaller. We have units which have up to 8 vessel capacity hardware. And these units are used mainly by customers for things such as late stage screening, technology transfer, and also manufacturing support, for example, running satellite cultures. All of those different products, however, have certain common features. So they're all relatively small scale, which can lower the running costs for things like media or feeds. They all use single-use labware, which therefore makes it much faster to set up an experiment and turn it around at the end. As I mentioned also in particular, for example, for the Ambr 15 48-way platform, you have a very high throughput, which allows you to run many test conditions at the same time, particularly important, for example, if you need to run things such as factorial designs. It's important to note that when you're actually running these experiments, the Ambr provides continuous control and monitoring. So real-time measurement for pH, DO, temperature, and those things can be used to transfer information back to the control computers that run these systems and thus enable feedback control for things like gassing, feeding, etc., etc. Also important to note that these systems are all stirred, so they're not shaken like a microtiter plate. These are all mechanically agitated. All the systems also come with significant amounts of automation in terms of pipetting, pumping, and so on and so forth, which enables automated feeding and sampling capabilities. And that's assisted by the fact that all of the different types of Ambr, or the main types of Ambr, for example, the high throughput 250, as well as the 15 cell culture and the 15 fermentation, have the capacity for at-line integrated analytics, thus enabling feedback and accurate regulation of feeding. Finally, as I mentioned, they all come with the possibility for DOE software integration, which enables a customer to understand precisely how the key input factors can affect the curvature of a search space. I'd like to focus in particular in this presentation on the Ambr 15, and this product has been on the market now since 2010. And our customers invest in this system predominantly, as I said, for screening activities, in particular for screening CHO or HEC clones, screening certain types of media or feeds, and also, as I said earlier, a little bit exploring process parameters such as pH or DO. We've also more recently had customers who have tried to extend that boundary and have tried to use the Ambr 15 for things such as microcarrier cultures, insect cell culture, stem cell work, transient transfection, vaccine development, and even looking at the use of an Ambr 15 as a mimic for perfusion operations. And I'll come back to the perfusion comment a little bit later in this presentation in connection with the Ambr 250. The 15 cell culture system will form the focus from now on, though, and it's a high productivity system that has 3 basic components. You can see on the left we have a small-scale miniaturized bioreactor. These are operated inside a robotic workstation, and those two are controlled continuously by a control computer software package. Focusing first on the workstation, so you can see towards the left we have a liquid handling robot which is used to remove caps from vessels. to remove lids from plates, and that is also used to pick up pipette tips. You can see towards the right-hand side we have the capacity to store either 1 milliliter tips, which can be used for adding feeds or adding media, or indeed for sampling. And we also have the capacity to hold 5 milliliter tips, for example, large volume, medium or inoculum additions. The reactors themselves are stored in blocks that we call culture stations. And on this platform, you can see we have a 24-way Ambr 15. And you can just about see in the middle of this diagram, I've highlighted we have 2 blocks of 12 bioreactors in each culture station. And one culture station can have 1 temperature and 1 agitation speed at any one time, but those values can be changed at any point during an experiment. All other properties, such as pH, DO, media strategy, inocular strategy, etc., those are all vessel specific. The media, the cells and the feeds themselves are held in plates, which you can just about see as these little white blocks at the front end of the robot. And once pipettes have been used, they are discarded automatically into the pipette tip bin. Although you can't see it, underneath the robot we have a pH and a DO card which scans every vessel once every 1.5 minutes and returns data back to the control software in order to enable it to take control actions. The robot is, as I mentioned, an automated platform, and this eases the use of the platform and enables good reactor control. So the Ambr 15 sits inside a biological safety cabinet. It sits there permanently, and the labware itself is single-use irradiated labware. Things such as caps and lids are handled automatically by the robot, and thus enabling automated feeding, base addition, antifoam addition, and so on and so forth. And this is true 24 hours a day, 7 days a week. So you don't have to be in the laboratory to carry out these actions. The robot can do it automatically based on the instructions that you program into the control software. So just to give you, as I said, a few key examples of control mechanisms, we have pH and DO sensors at the bottom of every vessel, and those pass into control loops running on each individual bioreactor. So in terms of pH, for example, in the Ambr 15 software, you would assign a pH set point and effectively a deadband for every vessel. If the pH rises too high, then carbon dioxide would be brought in to bring the pH down under the control of a PI controller. Similarly, at the bottom end, if the pH is going too low, then base will be added automatically to push the pH back up again. Equally, for dissolved oxygen, we have the capacity to sparge in or extra pure oxygen in order to maintain a DO set point, again under the control of a PI regulator. The bioreactor itself has a number of features which enable it to mimic much larger scale vessels. So at the top we have a vessel cap, which is designed for reliable robotic removal and replacement. Inside the vessel we have a sparge tube. Actually, we have 2 different versions of a vessel, one with a submerged sparge tube that terminates near the bottom, and another one which has a headspace gassing inlet that pushes gas in purely into the headspace. And you can use that vessel in particular, for example, if you don't want to use antifoam within your process. Either type of vessel can be operated at the same time on an Ambr 15. As I mentioned, this is a stirred system, so we have a pitch blade impeller located near the base of the vessel. And right at the bottom, underneath, you can see we have 2 spots, one as a DO sensor and one as a pH sensor. So all of these features enable us to operate the Ambr 15 in a manner that is representative of a much larger scale bioreactor. And that's all regulated through a single control computer software package, which enables us to create process recipes. You can see an image of that towards the top right of this screen. As an experiment continues, we have full visibility on the data that are being generated and the control actions that have been taken. So we have an audit log, which gives visibility of all events that took place, and enables us to track in real time the data such as pH or DO. And we have software which enables us to compare reactors both within as well as between experiments. So you can determine batch-to-batch consistency, if you so wish, between successive Ambr 15 experiments. And that all enables you to evaluate many conditions at the same time. As I mentioned, the culture station, i.e. the block of 12 vessels, provides one temperature and one RPM at any one time, but values can be changed whenever you want. And the properties can be varied between the stations. So if you have a 48-way Ambr 15, you could have 4 different temperatures or 4 different agitation speeds operating at the same time. All other properties are vessel specific, so different media, feed strategies, inocular, and so on and so forth. 2 different versions, as I mentioned, a 24 and a 48-way. The 48-way version gives you a very high throughput and gives you, therefore, capacity to obtain significant amounts of process knowledge, in particular at a very small volume, which reduces the footprint and also the cost per test condition. It's also very quick to set up. Setting up, say, a 48-way with all the bioreactors, putting those into the stations, can take no more than a few minutes. And that can all result in a dramatic rise in bioreactor throughput. So I have a little scenario here. So if we assume that you had one operator running a 14-day culture, and if you assume that that one operator could run either 6 2-liter benchtop vessels or a single Ambr 15 24-way system, or a single Ambr 15 48-way system, and making certain assumptions for turnaround and number of working weeks within a year, you can see that in the 6 bioreactor benchtop case, that person would be able to run approximately 84 bioreactors. If you switch that to an Ambr 15 24-way, you get over 500 bioreactors, and obviously for a 48-way that doubles and gives you the capacity to run over 1,000 bioreactors in a single year. We've also got capacity, as I mentioned, to have at-line analytics. So from very early on with the Ambr 15 cell culture, we integrated cell counters such as the Vicell XR or the CDEX high-res for calculation of viable cell density, cell viability, as well as all the other standard properties that cell counters measure. And that can be used in feedback loops, for example, to initiate liquid additions based on the viable cell count. We also have a system for at-line pH recalibration to improve control of vessel pH. And most recently, we've announced that we can integrate a metabolite analyzer, namely the Nova Biomedical Bioprofile Flex 2, for measurements of typical metabolites used in cell cultures such as glucose, glutamate, glutamine, and so on and so forth. And that can enable closed feedback loop control for topping up certain critical components such as carbon sources. And the final element of the Ambr 15 is the DOE software. So we integrate the Umetrics Biopat Modder Ambr software. With this, a user can choose the suitable input parameters, such as the type of media, the volume of media, the type of cell line, etc., etc. So you can use both qualitative as well as quantitative factors within analyses for DOE within the Ambr 15. And we provide an interface between the main Ambr software and the DOE package, which enables a user to generate and execute the Ambr runs. And then the output data from an experiment can then be entered into Modder and analyzed to map the search space. So now what I'd like to focus upon is some of the data that customers have been able to generate. This is one specific case study which is reproduced here with permission from the authors shown on the slide. And actually this is a free to download open access paper, so you should be able to get this directly from the internet. This was a 2-part study. In the first part, they used 2 monoclonal antibodies and tried to compare the Ambr 15 with a 2-liter benchtop bioreactor, specifically for viable cell density, percentage viability, titer, and product quality. And then in the second part of the study, they took another monoclonal antibody and did a DOE optimization on the Ambr 15 cell culture, and then tried to scale up that optimum to 2-liter, in this case trying to optimize pH and DO. So the first part of the study was, as I said, trying to demonstrate comparability, and they showed that the Ambr 15 matched the 2-liter bioreactor. So these are quotations that are taken from the authors, so these are not my words. They say that there was overall consistency in cell density and viability. Protein production followed a consistent trend between Ambr and the 2 liters. And if you look on the graph on the right-hand side, what we have there are certain product quality attributes. At the top, we have HPLC, reverse phase HPLC and size exclusion HPLC data. And towards the bottom, we have ion exchange chromatographic data. And the bars in each case represent the outcomes from the 2-liter bioreactors and replicate Ambr 15 vessels. And you can see that the sizes of the bars are very similar between the benchtop and the Ambr 15. And so they said, importantly, product quality attributes indicated comparable protein quality profiles between the different scales. In the second part of the study, they did a DOE optimization, in this case using JUMP, but in our case we use the Umetrics model package. And this is a 3-parameter multivariate plot. So the way to interpret this is to look for the intersection between the pH or the DO inputs and the output, which in this case was protein titer. So in this case, for example, you can see there's a strong correlation between the pH on the x-axis and titer on the y-axis. Up to a pH of 7, they were able to obtain a maximum titer of 3.3. And I'll come back to that value in a few moments' time. They also showed that when they compared DO versus titer, there was no significant correlation. So DO doesn't appear to have any critical impact. So they decided for subsequent studies to fix on the midpoint, i.e. 30%. They also did some further analysis looking at the effect of varying pH on things such as product quality, product quality, and also integrated viable cell density, CVC. And again, they showed that increasing the pH gave rise to a higher QP and a higher CVC. When they then scaled up from the Ambr 15 to the 2-liter bioreactors, if we look at the top 2 graphs, here we look at pH as an impact on the CVC, the cumulative viable cell density. And you can see towards the end of the 14-day experiment, the final output achieved on the Ambr 15 as a result of the DOE matched that obtained on the 2-liter benchtop bioreactor. Similarly, if you look on the graph towards the bottom right, which shows titer, it shows that the outcome achieved on the 2-liter reactor again was about 3 to 3.5, very close to the value of 3.3 that was obtained on the Ambr 15. So this suggested that we can use the 15 as a good DOE model to enable us to predict the outcomes for much larger scale vessels. And what that effectively means for our customers is that because we have continuous process control, we can achieve high quality, very rich data sets to aid decision making. And that can increase upstream productivity, especially because we have same-day turnaround capacity. And this can enable customers to make better use of employee time. They can spend more time evaluating output data or determining which experiments to carry out next. And it also means that if there are failures, for example, if you identify a specific medium is not particularly good, or if a particular pH turns out to be suboptimal, you can write off that failure far more quickly and far more cheaply than you might have been able to do if you were running, say, a benchtop vessel. So that's the Ambr 15 part of the presentation. I also very briefly wanted to talk about the big brother, so to speak, the Ambr 250 workstation, which, as I mentioned earlier, is intended for development, characterization, and optimization studies. This unit actually comes with its own equivalent Class 2 biological safety cabinet, again with software control, and in this case in either a 12-vessel or a 24-vessel bioreactor capacity. Unlike the Ambr 15, on this system we have additionally 4 pumped lines for every reactor, but we still have the liquid handler for aseptic pipetted additions and for sampling. And we also have stirred deck positions which can hold either 1 litre or 175 milliliter bottles for things such as feeds or for media, and additionally chilled or frozen sample storage and feed storage capacity. The 250 high throughput is a fully featured miniature bioreactor, so this is geometrically similar to much larger scale vessels, and we have a large number of features here which make it comparable to a larger bioreactor. So an exit gas condenser, a standard pH probe, gas inlets for both sparge and headspace gassing, so both are possible in this reactor simultaneously. We also have capacity to draw the off-gas out to an external off-gas analyzer if you wish for microbial customers. We have sparge tubes as well as headspace gassing inlets and a twin impeller. In the case of the mammalian vessel, which you see on the left-hand side, we have twin pitch blade impellers, whereas the one on the right-hand side for microbial, we have twin Rushton turbines. And we also have a vessel designed specifically for other types of operations, and some customers use that, for example, for microcarrier cultures. 250 technical specification is such that we have fully independent bioreactor control for all properties this time, including agitation speed and temperature, as well as all the other things such as pH, DO, off-gas, pipetting, pumping, and so on and so forth. And the effect of that is shown as an example here with some limited quantities of data which I've taken from a customer presentation. In this case, for a mammalian cell growth, titer, and product quality profile plot. So we have here one particular cell line. I haven't got the exact details, but you can see in the two graphs on the left-hand side, which shows a viable cell density and cell viability. We compare here 4 3-liter reactors versus the Ambr 250 high throughput. You can see a very good comparability there. And equally the same story for the antibody concentration, which is shown in the plot on the right-hand side. And although it's a little difficult to see, if you look at the table at the bottom, which shows some critical glycosylation information, you can identify very close matches between the Ambr 250 and historical 3-liter vessels. So good comparability for growth as well as for product quality details. And that enables very high throughput process development, remembering in particular that we have DOE, and you can see a little example there for a DOE that was executed in a benchtop vessel and also on the Ambr 250 high throughput. Just looking at the plot, you can see a good comparability there as well. As I mentioned, very briefly, I also wanted to talk quickly about the perfusion system. So we've developed a perfusion option for the Ambr 250 high throughput. So the high throughput system is now fully capable of operating in both fed batch as well as perfusion modes. The perfusion labware is capable of supporting the typical range of perfusion options, so either, say, an intensified fed batch or a true perfusion operation. The vessel includes a single-use perfusion filter, and we have a high-efficiency sparger for supporting high cell densities, and the capacity to enable medium exchange of up to 4 volumes per volume per day. And with the system, we would also supply single-use bags for collecting bleed and permeate. And that system is actually available to purchase from now on. And very briefly, I just wanted to show one quick data slide. This was data taken from a 2-reactor test where we specified target viable cell density, and that, along with the growth rate, were entered into the software, and the bleed volumes were calculated automatically by the Ambr 250 and executed. And we compared a single Ambr 250 versus one 100-liter bioreactor control. And you can see there's a reasonably good match between the viable cell density in the two scales. This was a very early test, so there was a little bit of variability, but we're still working to improve and generate better quality data. But you can still see there's a pretty good match there between the two scales. There was also a good match in terms of titer and metabolites between the scales as well. So that's pretty much it. I just wanted to give a brief conclusion. As I said, both the 15 and the 250 systems can deliver good agreement with much larger bioreactors. They're both automated parallel bioreactor systems which deliver high consistency and high throughput and create a step change in the capacity within a laboratory using the same FTE capacity and the same, and in fact using less laboratory space. And this can create a very cost-effective solution to enable large-scale studies in a very short space of time. They're both ideal models, in particular for the 250, as scale down for screening, development, and optimization. So finally, I'd just like to acknowledge the development staff here at TAP Biosystems, as we used to be known now, Sartorius, the development and the support staff, as well as my sales and technical colleagues, our customers, especially for sharing the data, and of course to you, as well as also to Biopharmacia and for Stephen for hosting this particular presentation. And if there are any questions, I'd be very happy to take them. Thank you very much for your time.
Thank you, Sunil. Now we will begin the question and answer segment of the webinar, but once again, I'd like to remind the audience that you can still send in your questions via the questions tab below the webinar screen. Now on to our first question. What's the next and future direction for microbioreactor application in WuXi Biologics? And where do you go from here?
Okay, I will try to answer this question. It's Liang here. So we're already seeing this microbioreactor is a powerful in the process, in cell culture process development. At the meantime, I think most of the application is limited to upstream process development, including the new model we just hear from Sunil about this perfusion Ambr 250. For the future, we do like to see microbioreactor can link it to the downstream process development. The limitation right now in our hand, or we're still working on that, is the volume. For the 250 ml microbioreactor, the volume is 250. For the traditional downstream process development, or the media or assay process development, we need quite a lot of material. So right now we're still going back to do 3 liter or 15 liter benchtop bioreactor production. So we think more work needed to bridge the upstream and downstream, and then can speed up the process development and then provide enough material for the downstream process development. I think that's something I think will come up in the maybe next year or next couple of years.
Thank you. Our next question. If my company has several different Ambr systems, 15, 250 HT and 250 modular, and we wish to compare the data from multiple experiments on those units with one another, can we do so?
Yes, you can. So we provide a particular piece of software called the results viewer, which enables customers to look at multiple types of Ambr experiments at the same time. So you can compare the outcomes from Ambr 15 cell culture, Ambr 15 fermentation if you want, the 250 modular and the 250 HT at the same time. And that piece of software is actually available freely. So anyone can look at that at multiple sites within a company spread geographically around the world, and that makes it very easy to compare and contrast the outcomes from different scales of operation.
Is it possible to run both mammalian and microbial experiments with the same Ambr 250 high throughput hardware?
Yes, it is. So the basic hardware is essentially the same in both cases. So a specific unit can be used to run a mammalian system, a mammalian culture, as well as a microbial culture. It would be advisable or sensible to have a good sterilization process in place to enable that to take place. But I have known customers that have actually got this exact scenario where they have a suitable facility design set up in such a way that they have access to both mammalian and microbial cultures running sequentially, one after the other, on the same system. The hardware is capable of supporting both types of applications. So yes.
Thank you again. Our next question is, what's your view of the microbioreactors as scaled-down models?
So I can answer this. This is Jimmy from WuXi Biologics. We do see a lot of potential for using microbioreactors, for example, the Ambr 250, as scaled-down model for both late-stage process development as well as for process characterization. As indicated from the presentation, we have started to implement that for multiple projects, and I do see a tremendous benefits and advantage of using microbioreactors for those purposes. Also, in WuXi Biologics, we extensively use single-use bioreactors at our GMP manufacturing. So having something like a disposable scaled-down model is also a need for us. As Dr. Tang showed, some of the cases we do see issues from using the benchtop bioreactors as scaled-down models. Those cases, we believe that the microbioreactors might offer a better scaled-down model advantage.
Thank you for that. Our next question, do you usually face contamination problems whilst using Ambr 15?
Not in my general experience, no. Standard aseptic technique is more than adequate for operating an Ambr 15. If you have serious problems with contamination, you could always subject the system to a vapor phase hydrogen peroxide rinse. But in general terms, no, I don't tend to find that. We can sterilize the liquid handler using peroxide, liquid peroxide. We can wipe the system down with alcohol, and provided you're careful with standard aseptic techniques, I haven't found any specific issues with contamination. Where I have had contamination, in a number of cases, it has been because things such as media or such as feeds or such as inocular that have been provided to me themselves have been contaminated. But provided you are, as I said, careful with aseptic technique and avoid those types of issues, no, Ambr 15 doesn't generally tend to suffer from contamination problems.
Yeah, our experience also indicated that we do not get higher contamination problem with Ambr 15.
Thank you. That's good to hear.
Our next question is, can I run different 250 experiments at the same time?
Yes. So the way the Ambr 250 software works is that you can write as many protocols as there are bioreactors on the system. So if you have a 24-way Ambr 250, you could create up to 24 completely different experiments running at the same time. And in theory, those could have 24 different sets of feeds, different sets of inocular, different sets of media, and they will run in parallel, but fully independently of one another. So that gives you a lot of flexibility with the 250. That's true for both the 250 high throughput as well as the 250 modular systems.
Thank you. Our next question is, do I have to run, for example, all reactor positions in a high throughput system?
No, you can run as many or as few as you want. If you want to run just one reactor, you can run that. If you want to run up to 24, you can do that. And even you've got the capacity if you want to start one experiment, say, I don't know, say you have a 24-way Ambr 250 and you start all 24 reactors, and let's say one of the reactor positions finishes early because maybe it was a shorter culture, you can stop that particular culture, and you can put a new vessel into place and start a new experiment in that position while the other vessels continue to run their old experiment. So you have a lot of flexibility in the way that you can set up runs on the Ambr 250.
Thank you for your answer. I believe, unfortunately, that's all the questions we have time for, but thank you for sending them in. Before I finish the webinar, I would like to ask our presenters, Dr. Li, Dr. Tang, and Sunil, if they have any closing remarks.
Yeah, I think there's another question coming in.
Oh, just one more question, though. Sorry. We can squeeze that question in. Since Ambr 15 geometric design is not conventional, did you see inconsistency, if any, in the scale-up from Ambr 15 to larger bioreactors?
No, because although an Ambr 15 is not geometrically similar, the mixing conditions and the capability to have at-line monitoring for pH and for DO as well as temperature control means that we're able to achieve a microenvironment inside that bioreactor which is able to give results which are consistent with the outcomes that you see in much larger scale vessels. And the best way for me to answer that particular type of question is to show the data. So if you look at the data that we've generated from our customers or from our developer partners, we can get very good consistency between 15 mil scale and say tens of liters, or even in some cases hundreds or even thousands of liters if you really want. And so you wouldn't be able to obtain that if the differences in the geometry were causing significant problems in terms of the growth model. So no, I don't see that as generating problems in terms of scale-up. Thank you.
Thank you. Thank you for that. Dr. Li, Dr. Tang, did you have anything to say on this?
Yeah, our experience is also that we don't see any issue in the geometrical design. Although there's also Ambr 50, which sometimes offers some added advantage for scale up. There are some, subtle differences in terms of the, for example, the volume ratios for spargings, as well as some probably sublocal mixing condition differences. So we would encourage that customers perhaps try both the Ambr 15 and Ambr 250 to see for their particular case which one might be a better scaled down model.
Thank you for your answers. That concludes the question and answer session. Once more, I'd just like to say, if you do have any closing remarks, would you like to say them now?
Thank you, Stephen. I think from WuXi Biologics perspective, we do see tremendous opportunity as well as advantage of using microbioreactors for all steps of the process development, from clone screening to process optimization to upscale down, as well as for late stage optimizations. Also, as mentioned, because a lot of our manufacturing are based on use bioreactors, so using a microbioreactor that's single-use nature also offers some advantage as a scaled-down model to us.
Yes, I would echo those comments fully. So in the case of our customers, we see many people using both the Ambr 15 and the Ambr 250. The 15 is used predominantly for earlier stage screening, the 250 for later stage work, but both have the capacity to predict much larger scale outcomes. And just as a final comment from me, one of the things that was raised earlier was about the fact that you need to start to integrate smaller scale downstream models as well. So this is an area within which Sartorius has been active. And indeed, there is a new type of product which has been developed and launched called the Ambr Crossflow, which is a small scale filtration system which has a very small working volume and very small cassettes with a high throughput capacity, which is intended for being compatible with the very small volumes which come out from microscale bioreactor systems. So there is a lot of work that is going on within this area as well.
Thank you all for your comments. So if you'd like to say something, go ahead.
Okay, I'd just like to add in, I'm glad to hear there's new effort to move an Ambr system with the downstream. I think all the technology which improve efficiency or provide flexibility and then keep the quality, the peculiar high quality of product into the system and really or speed up the whole process in biologic process development, and then from DNA to IND submission or to the late stage BLA submission. I think I'm looking forward to see this new product coming out and to learn more about their applications. And thanks, Stephen, for organizing this seminar.
Well, thank you. I just want to take this opportunity again to thank our presenters, Dr. Li, Dr. Tang, and Sunil, for sharing their knowledge with us. And I'd like to remind our audience that they can view this webinar and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. Thank you, everyone, and have a great day.
Thank you. Bye.